We are at Kansas State, and we are in Manhattan, Kansas, in the Little Apple, not in the Big Apple. And we are switching topic now. So we talk about animal models for COVID-19, and in this case, companion animal models. And the work we do can only be done in a facility which has BSL-3 egg space, so space which we can work with this agent in a safe and secure manner in large animals. In larger animals, cats, we did also the pigs, etc. So what is the rationale to do a companion, in this case, a cat model? We all heard that the potential reservoir is most likely bats of this virus of COVID-19, SARS-CoV-2, and then either via pangolins, as an intermediate host, or directly from pangolin, it spilled over to humans and caused the epidemic and then pandemic. We also know that there are natural infections, not only in humans with SARS-CoV-2 infections, but also in cats. We had an outbreak in a zoo here in the United States, and in the Bronx, in tigers and in lions. And then we in house cats, domestic cats, and in dogs, and we have outbreaks in mink farms, which are farmed for production of their fur in various countries, mainly in the United States, the Netherlands, and Denmark. We had outbreaks. The biggest production facilities of mink are in in China, but we haven't heard about an outbreak in China. But we know about the Netherlands, Denmark, and the US. And we also know there is one paper posted that there is a possible mink to human, a reverse zoonotic transmission from mink to humans. But these detection in SARS-CoV-2 naturally infected animals is rather limited, considering the extent of the human pandemic. So we did 2 cat experiments in the past 4 or 5 months. We are not coronavirus people, but we are zoonotic disease, infectious disease people. And I explained the timelines and how we do that here in this schematic. And this work is mainly done by Natasha Godreau and Daniel Madden. So we use cats, as we have never done cats before this event. So we use 2 cats as control animals for necropsy to have tissues and whatever, control samples. Then we used 6 cats in 2 groups, which were 5 months old, and we infected them with a 10 to the 6 TCID50 of the US strain, the WA1 strain of COVID, simultaneously in the nose and per os. And one day after this primary infection, we added 2 animals, 1 per group. We had 2 groups of 3 cats, 1 per animal as sentinel naive animals. These were naive cats. And then on day 4 and day 7, we necropsied 2 cats to see how the virus distributes through the body. And we took a lot of samples, as I show you in the middle, in a minute. And then on day 21, we necropsied 1 out of these 6 cats to have a day 21 sample. And then we reinfected the leftover 3 cats with the same amount of SARS-CoV-2, again intranasal and per os. We followed these animals for 4 days, post-secondary challenge, and then necropsied these animals. So this is how we house these animals in BSL-3 egg. So they are housed in group housing, 4 animals per cage. These are the 2 cages here on the left, and on the right, you see before when we treat them, bleed them, etc., we put them in single caging. And here's the treatment table. And we also do a lot of environmental studies, including air sampling, which I don't have time to go into today, but this will be posted very soon on or published very soon. So what is the sampling time of this primary infection? So as I mentioned before, and in red are the infected animals, in blue throughout are the sentinel animals, and the controls are in gray. We infect these cats on day 0. On day 1, we add the sentinel animals. We do body temperature, clinical signs, and body weight at various time points. Daily, the first 1, 2, and body weight at 2-3 days apart at different time points post-inoculation. We take nasal or pharyngeal and rectal swabs on various days post-infection. We do a lot of environmental sampling. We do blood collection as shown here. And then we did necropsies, as I mentioned, on day 4, 7, and 21. And we, at necropsy, we also do nasal washes. And we take bronchoalveolar lavage fluid, BALF. We take the entire respiratory, include upper and lower respiratory tract, GI, gastrointestinal tract. We take central nervous system, various nerves, major organs, etc., etc. When we looked in the blood, the controls are negative. None of the red are the primary infected animals were positive for RNA, and none of the sentinel animals became positive for RNA. When we did nasal swab analysis, in red, the infected animals, already at day 1 post-inoculation, we see high amounts of viral RNA. These are copy numbers at day 1, and they are maintained up to day 10 post-inoculation, are negative on day 14. Sentinels in blue come up 2 days later. We add them at day 1, contact co-house them, and we find positives already on day 3, and they are maintained also positive throughout day 10, and they are negative on day 14. We do nasal washes, and nasal washes we do at times of necropsies. We did 2 animals on day 4 and 2 animals on day 7 of the primary infected animals. They have high amount of viral RNA. Nasal washes at these days and are negative on day 21. Similarly, if we take bronchoalveolar fluid, we see day 4 and day 7 post-inoculation positive, day 21 negative. Or pharyngeal swabs, we see a similar picture as shown here in red and blue with the sentinel, starts on day 1 and maintained until day 10 in the primary infected animals. In blue are the sentinels, and they are positive already 2 days post co-housing, co-mingling, and are positive until day 5 and then negative. Rectal swabs, the story is a little different. The kinetics in red, you see the primary infected animals, they are positive on day 3. And they have very high levels of RNA maintained until day 14. So they are a little longer positive, these animals. And when we look at the sentinel animals, they have a similar kinetic as shown here. So we did the necropsies, and on necropsies, we did day 4 on the lower end. You see day 4 in orange, 2 animals, day 7 in red, 2 animals, and day 21 in purple, 1 animal. And here's the summary of the upper respiratory tract, nasal turbinates, soft palates, trachea, and you can see these animals are all positive, tissues are all positives on day 4, 7, and day 21 post-inoculation in all animals. If you look in the lower respiratory tract, we see positivity on day 4 and day 7, but the majority of lung lobes are already negative on day 21. Only a few are still positive. If you look at lymphoid tissues, lymphatic tissues, in tonsil, various lymph nodes, and the spleen, you see the lymph nodes and tonsils are positive on all days, in all animals. The spleen is not positive on day 4, but becomes positive on day 7, and it's maintained through day 21. If we do the same with other tissues like the GI tract, gastrointestinal, liver, heart, etc., what's listed here, you see most of these tissues are positive already in most animals on day 4 and day 7, and some become negative by 21, like the GI tract pool, the liver, the heart, etc. We did histopathology. We did it in the nasal turbinates, trachea, and the lung. And here I show an example of the bronchi. And you see here on the upper left, the mock infected animal bronchi. On the upper right, for day 4, on the lower left, day 7, and on the lower right, you'd see day 21 post-inoculation. And what you see on day 4 and 7, you see infiltration in the submucosal glands, adenitis, lymphoid cells, cells in the submucosal glands of bronchi. We see the same in the trachea. And when we look by cytohybridization, that's on the right here, On the upper panel, the mock, on the middle panel, the day 4, day 7, and the lower panel on day 21 post-inoculation. And we see on day 4 and day 7, we see viral RNA in these glands, submucosal glands. And if we do immunohistochemistry with the antibody against the N protein, we see also viral antigen in the same lesions. If you look for neutralizing antibodies, these are the animals which survived past day 7. On day 10, all of these animals, the sentinels as well as the infected animals, have neutralizing antibodies. The infected animals, the primary infected animals, actually are already positive on day 7 post-challenge with neutralizing antibodies. And this is maintained until day 21. So this summarizes the first part, which is in press, which is actually posted on bioarchives, but it's in press at emerging microbes and infections. If we infect cats by the intranasal per route with the US strain of COVID, WA1, 10 to the 6 TCID 50 per animal, we did not find obvious clinical signs. Maybe a temperature spike at day 2. So this is a beautiful model for asymptomatic infections. As one of the previous speakers mentioned, most of human infection are asymptomatic. This is a beautiful model. However, and we do not see any changes. Actually, we did a lot of blood parameters. We have special machinery to do that in animals. We looked at all the blood, at all the blood cells, etc. We did not see any changes, except thrombocytopenia from day 5 on, and this is in the paper in detail. We do not see anemia in the blood. However, we see transient shedding of viral RNA through the oral, nasal, and rectal cavities. When we did necropsies, we found viral RNA regularly in the upper respiratory tract throughout the day 21, not only on day 4 and 7, but regularly throughout day 21 in the upper respiratory tract, lymphatic tissues, and in the olfactory bulb. And on day 21, there was not much left in the lower respiratory tract, GI tract, liver, and other organs. When we looked macroscopically and microscopically, we saw macroscopic lesions, but mainly the microscopic lesions we saw I didn't have time to show tropinates and nasal mucosa. We see rhinitis in the upper respiratory tract and in the trachea and bronchi. We see tracheobronchoadenitis with the presence of viral antigen and viral RNA on days 4 and 7, but not on day 21. We do not see interstitial pneumonia, how it's described, for example, in the hamster model of COVID or in humans. Very importantly, we see transmission to naive sentinels within 2 days of co-housing. So we still have to finalize our virus titrations and sgRNA analyses. We also do cellular immunity. We have the PBMCs and have the peptides for the NNS proteins. And we have done RNA-seq on these animals, and we have to analyze these. This study is the reinfection study, which is not published yet. It's in preparation right now. And we have done 2 of these reinfection studies in cats. And we used SARS-CoV-2 on day 21 post-primary infection. The first experiment, which I described first, was done on 5-month-old cats, and the second one on 7-month-old cats. This is the sampling time of the first experiment. On day 3, we reinfected these animals. Sorry, on day 0, we reinfected 3 cats with SARS-CoV-2, 10 to the 6 per os and intranasal. We followed them for 4 days, and we necropsied these 3 animals on day 4 post-secondary challenge. And we took the normal organs I described before. What happens if we look for RNA shedding in the nose and other upper respiratory tract samples? These are the primary infected animals and sentinel animals from the primary infection. If we look for in green for the reinfected animals, we see viral shedding on day 1, but it's negative on day 3 and 4. If we do the same with nasal washes at necropsy, we had 1 out of the 3 animals was positive with nasal washes at day 4 post-secondary infection. If we do the same with BALF, which is the lower samples, the lung, all 3 animals are negative. If we look at tissues, we also did oropharyngeal swabs and rectal swabs, and the rectal swabs were positive and the oropharyngeal swabs were negative in these reinfected animals. If we look for RNA in these various tissues, in this case, in this time, or with this graph, everything now post-secondary infection, so reinfected animals are in green. In the upper respiratory tract, most of the animals, the majority are still positive at day 4 post-reinfection. If we look at the lower respiratory tract, the majority is negative. There are only some few samples are positive. If we look in lymphatic tissues, and again, these are the green animals, the majority of the animals are still positive at day 4 reinfection. And if we look at other tissues like GI tract, etc., you see some are positive and some are negative. GI tract is negative, CSF majority is negative. If we look in the trachea and the bronchi, and we were very concerned about enhancement, not only about how this primary infection protects against reinfection, but also the primary infection can enhance disease. And we look very careful in all the tissues, trachea and bronchi are demonstrated here. On top are the mock-infected tissues, on the left H&E, and the other one is inside of hybridization. And you see very nicely on day 4 post-primary infection, you see the submucosal adenitis, and it's associated with RNA in the lesions. The same is true for bronchi and for trachea. If we looked at day 4 post-secondary infection, we did not see any lesions, pathological microscopic lesions, and no antigens or RNA, neither in the bronchi nor in the trachea. The second reinfection experiment was a little different, and it's described here. We reinfected 5 cats, 3 primary infected, 2 sentinels. Sentinel is more natural infection. And then we added 2 sentinel cats, naive cats, 1 day post-reinfection into the room to see whether shedding of these reinfected animals can lead to reinfection of naive cats. And as you know, they're highly sensitive to transmission, so cats transmit very well. And then we did the whole 9 yards of sampling of everything, blood, we did clinical analysis, and then we necropsied animals again at day 2, 2 animals at day 4 post-secondary infection, and 2 animals at day 7 post-secondary infection, and then 3 animals at day 14 post-secondary infection. We wanted to see whether the sentinel cats will seroconvert. What did we see in the upper respiratory tract? Similarly as before, we saw shedding of the virus on day 1, but also on day 3 or 4 post reinfection. The sentinels did not shed throughout the entire observation period. In this case was only 14 days. The blue and red are the primary infected and the primary sentinel animals. If we looked at nasal washes, on day 4, we didn't find anything, but on day 7, both animals were positive, and all 3 on day 14 were negative of post-secondary infection. If we looked in BALF, all animals in the lower respiratory tract, all animals on day 4, 7, and 14 post-secondary infection were negative. Importantly, we looked whether there's an anamnestic immune response, and let's focus on day 21 post-challenge, which is day 0 post-secondary challenge. And in brown, in the brown background, you see the animals which were reinfected, these 5 animals. They all had titers between 40 and 320 of neutralizing antibodies. At the time of necropsy, day 4, day 7, and day 14 post-secondary infection, 4 out of these 5 showed an anamnestic immune response. Importantly, in blue, the lower 2 animals, the sentinel animals, they were negative at the time when they were co-housed with the reinfected animals. However, when we when we looked at day 14 post-secondary infection, their antibody levels, neutralizing antibody levels were still negative. So there was an anamnestic response in the primary reinfected animals, but there was no seroconversion in the sentinels. So this brings me to the summary of my second part of the study, and this is in preparation, reinfection of cats. If we reinfect primary infected cats, we do not see any obvious clinical signs. We do not see changes in blood cell parameters, biomarkers, and no anemia. However, we see RNA shedding mainly via the nasal cavity, but also in one experiment via the oral cavity and in the rectal cavity. We see RNA nasal washes, upper respiratory tract on day 4 and 7, post-secondary challenge. We see also significant RNA levels in the upper respiratory tract of these reinfected animals, not in the lower respiratory tract on day 4 post-reinfection, in the lymphatic tissues and various organs. And importantly, we didn't see an enhancement of respiratory disease. We didn't see microscopic pathology, and no antigen or RNA in lung tissues at 4 days secondary challenge. We are still working on our second experiment on the pathology on that. So this will be summarized in the next couple of days. Importantly, we saw an anamnestic immune response after reinfection at day 7 and 14 days post-secondary infection. At day 4, in the first experiment, we didn't see that. We have to wait apparently a little longer to see that, which indicates that there are seems to be some replication going on a minor level. However, very importantly, there was no transmission and seroconversion of sentinels, which were co-mingled with the reinfected cats for 13 days. So the virus, whatever they shed, is not enough to transmit to sentinel animals. So our conclusion is that SARS-CoV infection in cats induced humoral and most likely cell immune responses, which provided pretty good protection against reinfection, however, not mucosal immunity in the upper and GI tract, upper respiratory and GI tract. So immunological responses to prevention and treatment of SARS-CoV-2 are possible. This is pretty not so easy to work with these animals in BSL-3, so it takes a village. So many people in my laboratory, and here I have pictures of the people involved: Bianca, Natasha, Daniel, David, and Jesse. We also had help with Wenxu Ma, who is now at University of Missouri, Bill Wilson from USDA, and Adolfo Garcia Sastre from ICANN School of Medicine, Monsanto, and the immunohistochemistry and cyto was done at LSU with Udani and Mariano. The work is very expensive. One day in BSL-3 is about $1,000, so you need money to do that, and so I'm very grateful to my funding organization. We started with internal funds from Kansas State and from the state of Kansas, and now we got grants from NIAID and DHS. Thank you very much for your attention.
Thank you, Jürgen. We have a question from Neville Kleins. Neville, you should be able to talk now.
Yeah, hi, thank you. Given that in the reinfection study, you didn't have any transmission to the sentinels, and also there's little or no histologic evidence. I was wondering whether you correlated the RNA levels with any sort of virology culture results. Is it possible that you're somehow stimulating release of cells that contain this RNA rather than viral replication, as we see sometimes in long persistent patients who are positive?
Yeah, this is obviously a good question. We have not done the virology. We are not done with the virology yet. And but even if we have positive virology and no transmission, what will it tell us if I find 10 to the 1 TCID50 per mil and have no transmission? So I think that sentinels for me were very critical in the room, that they didn't seroconvert, because in all our studies we have done so far, there's very efficient transmission after primary infection to sentinel animals. This has been shown also in the zoo, in the New York Zoo, with the large cats. So, but we are doing these studies and we are doing environmental swabs, etc. So all this picture together, I only could show you at 25 minutes, only bits and pieces of what we do. Did that answer your question?
Yes, I mean, the word reinfection, I guess that's my question. Are they really being reinfected, or is it almost like a booster of a, let's say, a dead virus vaccine or something?
I think what we do is reinfection, because I use 10 to the 6 TCID50 and put it into the nostrils and into the into the oral cavity. So this is, in my opinion, it's called at least that's how I understand English, would be called reinfection. And what we do with it, the data we get out of it, is how I presented it to you. But I call this reinfection, and other people have published also and called it reinfection. So in my English vocabulary, it is reinfection.
Thank you.
Okay, we have a question from Brian Karolewski in the Q&A box that I'm going to go ahead and read. And it is, did you use SPF cats in this study, and would you expect any differences if the cats were seropositive for feline coronavirus?
It's another very good question. We have used SPF cats, and they were all negative, and we have discussed that in the paper. You can look it up in bioarchives. Yes, these cats were very clean cats. One cat was $1,500. Not cheap to get SPF cats. Yes, we used SPF cats. And they were not only SPF cats, but also clean in terms of antibody profile.
And I want to ask sort of a naive question. That is, are there reports of symptomatic cats in homes, or examples of transmission between cats and their owners in home environments?
Yes, that's the whole rationale, which when I started out, why do we do it? Because COVID-19 patients have regularly transmitted the virus to their cats, and some of these cats died, some got sick, and so there have been necropsies shown. Not only the isolated virus from these cats in COVID-19 households, but also have been necropsies to show that these animals get sick and have various histopathological lesions. And this has been happened not only in the US, but also in Europe, in Hong Kong, around the world. And there is a 15% seropositivity. They looked retrospectively in cats in Wuhan, and 15% of the cats they tested had positive serology for the SARS-CoV-2 specific antibodies.